Hi, my name is Hani Zirwatul Hanan binti Ishaq. I will begin this video presentation with the title Analysis of Acrylamide in Fruit Product using HLPC. High Performance Liquid Chromatography, HPLC, which is widely used to separate, identify and quantify chemicals, is basically an advanced form of column chromatography. HPLC is a form of column chromatography that pumps a sample that is dissolved in a mobile phase at high pressure through a column with a stationary phase. The properties of the sample and the solvent, as well as the nature of the stationary phase, determine the retention times of the analytes. As the sample passes through the column, analytes having the strongest interaction with the stationary phase will exit the column the slowest. It means they exhibit the longest retention time. Based on the article, we study acrylamide content in starch-based commercial food by using HPLC.
Assalamualaikum, my name is Muhammad Asad Abdullah. Today I want to present my section as methodology and conclusion. First, two different carbohydrate-rich foods were taken as samples, which are French fries, blended biscuits, and bakery biscuits. The ground sample and its extract were kept in negative 10 Celsius in a plastic screw cap centrifuge tube until analysis. Then, stock solution of 100 ppm and acrylamide are prepared and then dilute with ultra pure water to give several standard 10 until 1000 ppm solutions. All the solutions were processed at 4 Celsius for around 3 months. Whereas one solution was prepared in 100 ml distilled water by dissolving 15 grams of potassium hexacyanophrate, and whereas two solution was prepared in 100 ml distilled water by dissolving 30 grams of zinc sulfate. And then 1 gram of deflated sample was taken in the 15 ml of the centrifuge tube. Afterward, 10 ml water was added in the tube and centrifuge at 100 Celsius, 10,000 rpm for 10 minutes. The filtrate was transferred into another centrifuge tube. Then the filtrate was added with 0.5 ml of both Keras 1 and Keras 2 solutions. Mix again, centrifuge at 100 Celsius, 10,000 rpm for 10 minutes. The supernatant was removed and evaporated in the water bath at 400 Celsius for 15 minutes and filtrate with 0.5 μm cellulose acetate syringe filter paper. Then the analysis was done by Agilent 1260 Infinity model of liquid chromatography system. For the separation of the analyte, an isocratic elution pattern was adopted, and the water and acetonylate ratio 90 and 10 was used as the mobile phase. Thank you.
During an HPLC analysis of a mixture, the components will separate based on their retention times. This will produce a chromatogram like in the picture. Either the peak height or the peak area can be used to estimate the concentration. This can be done because these values are proportional to the concentration when the peaks are sharp and the flow rate is carefully controlled. For the qualitative analysis, the retention time tells us about the compound identity. Based on the experiment in the article, a standard acrylamide, which is A, was prepared and has the retention time of 0.464 minutes. Next, the prepared samples, which are bakery biscuit, French fries and blended biscuit, produced the peak at close retention time like the standard acrylamide under identical condition. The retention time of acrylamide in bakery biscuit, French fries, and branded biscuit in the experiment is 0.377, 0.481, and 0.472 minutes respectively. Based on the article, the variation in acrylamide concentration between several batches of a product category could be due to different processing conditions and chemical composition of the raw material used. The concentration of acrylamide in the prepared sample, which are the French fries, bakery biscuit, and branded biscuit, can be determined by using the peak area. The majority of modern chromatographic instruments nowadays come with computer or digital electronic integrators that enable accurate peak area estimation. Therefore, in the article, the concentration of acrylamide in the samples were gained, and the mean were calculated by using Excel. The mean concentration of acrylamide in the French fries, branded biscuit, and bakery biscuits are 929, 397, and 369 microgram per kilogram respectively. European Commission has recommended a value of 500 microgram per kilogram as indicative and 350 microgram per kilogram as benchmark level for biscuits. Overall, 62% of branded biscuits and 36% of the bakery section sample had acrylamide within an acceptable range, while the other remaining, which is 28%, had high content of acrylamide.
Next, in conclusion, based on the articles, the component which is acrylamide in the samples can be separated and identified by using HLPC. The component acrylamide in the samples was identified and the concentration was calculated by their retention time. The amount of the carcinogenic acrylamide pollutant in all of the sample is 1143.15 mg per kilogram, with the French fries having the highest concentration. In baked and fried food products which are popular in India, more than half of baked biscuit samples and all samples of French fries are acrylamide levels above the indicative value of European standards. It ought to worry regulatory agencies as well as industries. Aims should be made to prevent excessive browning, according to studies showing that the browning index can be utilized as a measurement index. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. My name is Arifam Azman, and now I will present about analysis of vitamin C concentration in commercial fruit juice by using UV visible spectrophotometer. Okay, for the introduction, UV visible spectrophotometry has been extensively utilized to identify and measure the concentration of organic molecules. The direct and derivatization techniques are covered along with qualitative and quantitative assessments. Additionally, various UV visible spectrophotometric detector types are presented, as well as how mathematical methods can be utilized to extract data from UV visible spectra. Okay, next, in the food sector, ascorbic acid is frequently used as a source of vitamin C or as an antioxidant. However, it quickly breaks down in beverages that have acidic pH and can accelerate the color loss of anthocyanins, which is a natural beverage color that causes the drinks to become colorless. Ascorbic acid is commonly added to drinks and foods to help replenish vitamin C content that may be lost in this process. To reinforce or add a citrus flavor, ascorbic acid is frequently added. Additionally, ascorbic acid serves as a preservative to prevent food from going bad, such as bread, cured meat, jams, and jellies. Now, I will explain about methodology. First of all, standardization of sodium thiosulfate solution. 5 ml of potassium iodide solution was pipetted into a conical flask. Then, 10 ml of 10% potassium iodide and 10 ml of dilute sulfuric acid was added to the conical flask. The liberated iodine with sodium thiosulfate solution was titrated from a burette until a faint yellow color is reached. Then, 10 drops of starch solution were added and the titration continued until the blue-black color of the starch triiodide complex just disappeared. Lastly, the titration was repeated until concordant results were obtained. Next part is titration of standard solution of ascorbic acid. First of all, 5 ml of the juice sample solution was pipetted into a conical flask. Then 10 ml of 10% potassium iodide and 10 ml of dilute sulfuric acid was added to the conical flask. The liberated iodine with juice sample solution was saturated from a burette until a faint yellow color is reached. Then 10 drops of starch solution were added and the titration continued until the blue-black color of the starch triiodide complex just disappeared. The titration was repeated until concordant results were obtained. The concentration of the ascorbic acid was calculated from the relation milli-equivalent of sodium iodide minus milli-equivalent of sodium thiosulfate equal to milli-equivalent of ascorbic acid. For the last part of methodology, preparation of standard calibration curve. First of all, standard solution of ascorbic acid containing 0 ppm until 30 ppm was prepared in 25 ml volumetric flask from 500 ppm stock solution of ascorbic acid. Each flask was added few drops of bromine water to oxidize the ascorbic acid to dihydroascorbic acid. A few drops of thiourea was added to remove the excess bromine and thus the clear solution was obtained. 1 ml of glacial acetic acid and then 1 ml of 2,4-DNPH solution was added to all standards and blank. All the flasks were kept at 37 degrees Celsius temperature for 3 hours in a water bath. Then all the flasks were cooled in an ice bath for half an hour and treated with 5 ml of chill 85% H2SO4 with constant stirring. The flasks were all added with distilled water until the calibration mark. The spectra was scanned to determine the lambda max for the complex form. The calibration curve were drawn at the proper wavelength. Now, we will proceed with the next presenter.
So I will tell about the analysis. This is the result and discussion estimation of lambda max, which is absorption maximum of ascorbic acid. Ascorbic acid standard solution at a concentration of 30 ppm were produced to determine the absorption maximum. And this is figure 1, shows the complex maximum absorbent lambda max, which was determined by scanning it in a range of wavelength from 350 nanometer to 650 nanometer. And this is the calibration graph, which is calibration curve in it. Calibration curve after determining the maximum wavelength of the colored complex 521 nanometer with a spectrophotometer. All standards absorbent levels from 0 till 30 ppm from a 500 ppm stock solution of ascorbic acid were measured in 35 ml volumetric flask. By tracing the concentration against the corresponding absorbent, the calibration curve was created in this figure 2. And ascorbic acid has a limit quantification LOQ of 0.017 ppm and a limit of detection LOD of 0.01 ppm. 3 out of 10 measurement of standard solution. For 10 measurement of the standard solutions, ascorbic acid concentration of 7 ppm, the relative standard deviation was 2.4%, and the calibration curve of the Ossezon's complex, which was created from a number of ascorbic acid standard solution, is shown in figure 2. So this is table 1, which is shown amount of ascorbic acid in packet fruit juice. So from the table 1, we note that there is a little difference between the amount labeled and the amount calculated. In all cases, the amount calculated was later than labeled. That is referred to unstable vitamin. So orange state the highest vitamin C. Lastly is conclusion. So in conclusion, vitamin C is present in a variety of local fruits, but not all fruits contain the same amount of it, because some have little vitamin C and others have a lot. In the current study, we discovered that every fruit contains biologically active photochemical. A quick and accurate method was the spectrophotometric method for evaluating vitamin C in fruits using 2,4-DNPH. That's all from me. Thank you.